Hello, everyone, and welcome to today's webinar, Exosomes, from Pure Isolation to Standardized Analysis by Flow Cytometry. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. For more information about our sponsor, please visit miltenyibiotec.com. Now, we encourage you to participate today by submitting any questions you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain those credits. Joining me now, I would like to welcome our speakers, Ariadna Pascual, a product manager for molecular analysis at Miltenyi Biotec, and Laura Müller, a product manager for flow cytometry reagents at Miltenyi Biotec. And joining them for the Q&A portion of this webinar will be Stefan Wild, a group leader at R&D and Miltenyi Biotec. Welcome to you all. You may now begin your presentation.
Welcome everybody to today's webinar on exosome research solutions. My name is Ariadna Pascual and I'm the Global Product Manager for Molecular Analysis at Miltenyi Biotec. We also have here my colleague Laura Müller, which is a Global Product Manager for Flow Cytometry, and Stefan Wild, Group Leader in the Research and Development Department, which will join us during the Q&A session. Okay, so let's get started with the webinar. This is today's agenda. First, I will give you a short introduction about extracellular vesicles or EVs, research and applications. Then I will introduce the different EV isolation methods with a focus on immunomagnetic isolation. After this, my colleague Laura will give an overview on EV analysis, specifically on our multiplex flow cytometry assay. Then she will show you data obtained with using our assays, followed by a short summary and a Q&A session. Okay, so let's jump into the topic. Extracellular vesicles, short EVs, are membrane-surrounded particles secreted by every cell type. Since the first observations in 1983, the interest in the research of extracellular vesicles have been continuously increasing, as shown in the chart illustrating the number of publications per year. In 2012, the International Society of Extracellular Vesicles was founded in order to advance and also to guide the research on these vesicles. And since then, the interest has been extended from fundamental research to translational and also to clinical applications. Just as an illustrating example, up to today, there have been more than 190 clinical trials involving EVs, many of them also investigating the use of extracellular vesicles to treat COVID-19. Okay, so what are exactly EVs and why are they so interesting? EVs are, as I mentioned before, membrane-surrounded vesicles that are secreted to the extracellular space from every cell type. They can be classified into exosomes and microvesicles based on their biogenesis. So while exosomes are generated by fusion of multivesicular bodies with a plasma membrane, microvesicles bud from the plasma membrane. And the size of these vesicles varies between 30 nanometers and 1 micrometer, being exosomes usually smaller than microvesicles. It has been shown that EVs are involved in cell-to-cell communications and they carry diverse biomolecules, including RNA, DNAs, proteins, and lipids, and they carry these molecules from the parental cell to the recipient cell. And the surface and also the cargo of the EVs will not only depend on the cell type, but also on the cellular metabolic status and on the physiological conditions. For instance, the composition of EVs secreted from a cell with a healthy state will differ from the ones secreted during cancer, for example, or during a different physiological condition like exercise. As shown in the illustration, tetraspanins are these transmembrane proteins broadly found at the surface of EVs and are commonly used to identify and also to separate EVs from complex samples. However, so far, there is no unique marker that allows to distinguish between the different types of EBs, and this is a major hurdle in the field since, as mentioned before, microvesicles and exosomes are very different, and the biogenesis, they are different. The biogenesis process, they are also different in the composition and may have also different signaling functions. Here we will talk a little bit about the different sources and current applications of EBs. As they are secreted to the extracellular space, as I mentioned before, EBs are present in all body fluids, including saliva, urine, or breast milk. Additionally, they can also be generated in vitro and collected from the cell culture supernatant of culture cells. Due to their non-invasive and easy accessibility, and their different composition, different profiles that mirror the physiological conditions of the parent cell, EBs have gained a lot of attention in different applications. For instance, they have been shown to be a very useful approach to investigate disease biomarkers, specifically cancer biomarkers, and they are also being investigated as delivery entities in drug discovery, but also in vaccine development, among other very interesting applications. After the overview about EB research and application, I will focus on the different technologies and most used methods for the isolation of EBs. As mentioned earlier, EBs are easily accessible in body fluids like urine, saliva, or plasma. Most of these sources where we can find these vesicles are highly complex and they are highly heterogeneous and contain not only EBs but many other vesicle cells, particles, and proteins. And a proper isolation will allow us to separate, clean, and also concentrate the EBs from all other components of the sample. And this is a crucial step for many downstream applications in order to generate reliable results. One of the requisites is purity. Isolation methods need to assure a high purity and a specificity, and this is particularly challenging when considered that EBs are highly heterogeneous in size and composition. So in this table, we find the most common use methods for exosome isolation or EB isolation. One of the most used methods is ultracentrifugation. These methods allow EB isolation from big volumes, and although it can be potentially, or it can lead potentially to high yields, it is very time consuming and one cannot be sure of the specificity of the isolated fraction, since all the other components with the same density will be also isolated. Like ultracentrifugation, other physical methods have the same disadvantages and can potentially co-purify non-EB components. Miltenyi Biotec has developed an assay based on immunomagnetic isolation, and this assay targets specific EB membrane markers which allow for a high specific and high purity exosome isolation. One of the big advantages of immunomagnetic isolation is also that it allows for the separation of a specific EB subpopulation based on different surface markers. Okay, so how does this method work? So this assay is based on positive magnetic isolation and use magnetic microbeads coupled with specific antibodies, anti-CD9, CD63, and CD81 that recognize and also they bind to the specific tetraspannings on the EB membranes. Here you can see the principle of the assay, and what happens is that first the sample containing the base is incubated with the magnetic or the immunomagnetic microbeads. Within the sample, EBs will be labeled with the microbeads containing or coupled to the antibody, and then the sample is then loaded onto a separation column placed on a magnetic separator. The magnetically labeled EBs will be retained within the column, while the unlabeled vesicles and other cell components, other contaminants and proteins will run through the column. So in the last step, the pure EB sample is eluted from the column by removing the column from the magnetic field. So as you can see, it's a very smart principle and a very easy protocol to follow. So here I listed some of the features and advantages of the exosome isolation kits. The kits use immunomagnetic microbeads in the nanometer range, and these nanometer or nano-size microbeads they always stay in suspension, creating an even distribution of the antibody, which allows for a much more efficient EV capture than other magnetic microbeads that tend to precipitate. The assay is fast and it's very easy, doesn't require special equipment. It can be used for virtually any EV source, including serum, saliva, plasma, urine, and from both human and mouse. Another characteristic of this assay is that it allows sample volumes as little as 0.5 milliliters, which I think it's very interesting when you only count with very limited material, which is what usually happens when working with, for instance, liquid biopsies from patient samples. Additionally, this technique has been proved to give excellent results in terms of purity, since it efficiently removes contaminants and isolates functional EVs regardless of their size. So here you can see the results of a study by Michael Pfaffel lab comparing different isolation techniques, where they show that compared to other isolation methods, immunomagnetic isolation exhibits higher purities, and it shows very clear bands in this protein analysis by Western blot for the common EB markers. So as I already mentioned, a pure EB sample, it is crucial for obtaining high quality and trusted results in downstream applications like, for example, flow cytometry, protein analysis by flow cytometry, genomic analysis, but also electron microscopy, for example. As mentioned before during the introduction of this method, the other big advantage of immunomagnetic isolation compared to the other isolation methods is that this method allows to perform subtyping. So it is able to separate different EB subpopulations within the same sample. And the physical separation of EBs, depending on their differences in surface biomarkers and in the profile or surface proteins, this has great potential and is winning a lot of interest in the field. For instance, it is a very interesting approach to consider in order to develop new assays to investigate, for example, cell type specific EBs or purify EBs released during a specific pathological condition. And just to finish with this part, I like to mention that this technique is compatible with many downstream applications, including electron microscopy, RNA sequencing, and also analysis by flow cytometry, which will be covered by my colleague in the next section.
Thank you, Ariadna. So after isolating highly pure EVs, you can of course perform whole exosome sequencing or microRNA sequencing to investigate the nucleic acid cargo of your EV sample. But in this section of the webinar, I would like to draw your attention to the protein cargo. And specifically, I'm going to present to you how to use flow cytometry to screen multiple surface markers on your EV sample. But before jumping into the topic, let's first have a look at the common methods used to analyze EV proteins. Different methods can be used to analyze EV proteins as shown here. These include, for example, Western blot, mass spectrometry, electron microscopy, and flow cytometry. Western blot is commonly used to demonstrate the presence of EV markers. However, it requires large sample volume and is very time-consuming. Then we also have mass spectrometry, which is more sensitive, but requires specialized setups as well as extensive operator expertise and also tends to be very laborious. And in both cases, it's not possible to analyze proteins at single EV level. Instead, single EVs can be analyzed by electron microscopy, where you can further detect proteins by immunostaining, but here only few EVs can be analyzed, and it's technically quite challenging. And another method to analyze single EVs is flow cytometry, which can also be used to analyze the protein cargo of EVs. Flow cytometry is a powerful method which is widely used in various research and clinical labs for high-throughput analysis. Unlike other methods, it enables the rapid measurement of multiple physical and biological properties of cells or particles as they pass in suspension through one or more laser beams. Thus, properties like size or immunofluorescence can be easily measured and quantified. And because of all these advantages, flow cytometry holds great promises for also being used to analyze EVs. Next to the flow cytometry itself, the most important component of a flow cytometry experiment comes down to the antibodies. Then by using highly specific antibodies, which are conjugated to fluorescent markers, it is possible to identify and quantify the target of interest. recombinantly generated reaffinity antibodies are designed and optimized for flow cytometry. They provide superior lot-to-lot consistency and purity due to standardized production, rigorous purification, and stringent quality controls. All reaffinity antibodies contain a specifically mutated human IgG1 Fc region to eliminate background signals. On top of this, they all have the same IgG1 isotype and thus only require one universal isotype control. This greatly reduces the complexity of experimental planning. And all the benefits recombinantly generated reaffinity antibodies offer come in handy when analyzing small particles like EVs by flow cytometry. Although flow cytometry is widely used in the field, its straightforward applicability to analyzing EVs is very challenging, and this is mainly due to the small size of EVs as illustrated here. So you can see the size of an EV is about 100 times smaller than the size of a cell. And small particles like EVs, they hardly scatter any light. And if analyzed by flow cytometry, you can see here in the plot on the right that signals are detected in the side scatter and forward scatter, but it's very likely that these are EV aggregates or swarms. Then single EVs, they would appear down here in the left corner, marked by the red circle. But this region is so heavily overlaid by noise, and often a trigger is used to avoid this noise, and therefore the EVs are not visualized. Still, it is possible to analyze surface markers on EVs by flow cytometry, but the co-expression of markers on the same EV is very difficult to address because, again, of the small size of the EV, it's hard to distinguish single EVs from EV aggregates, and on top of this, it's further hampered by the potential blocking effects of the antibodies. To overcome some of the mentioned limitations and to simplify the analysis of EV surface markers by flow cytometry, we developed a multiplex bead-based assay depicted here. How does it work? Well, the assay consists of 39 capture beads, which are labeled with varying amounts of 2 dyes to generate 39 distinct bead populations, which can be easily distinguished by flow cytometry. These are shown here on the left. And each of these capture bead populations is coupled to an antibody specifically targeting one of 37 exosomal surface epitopes or 2 isotope negative controls. Then after the incubation with an EV-containing sample, these capture beads are stained with a detection reagent, for example, a cocktail of APC-conjugated antibodies against the tetraspanin CD9, CD63, and CD81, which are commonly expressed on EVs. And then consequently, sandwiches complexes are formed between the capture bead, the EVs, and the detection reagent. And based on the fluorescence properties of the capture beads and the detection reagents, a semi-quantitative analysis of EV surface markers can be easily performed by flow cytometry. And the parallel detection of 37 different surface markers then results in a characteristic surface profile of an EV sample. Let's have a closer look how this would actually look on a flow cytometry. So as you can see in the first plot, with the side scatter and forward scatter gate, you can easily exclude the doublets and aggregates based on the bead size. Then with the PE and FITSI gate, as shown on the second plot, you can nicely discriminate the 39 different bead types based on the different ratios of PE and FITSI labeling on the capture beads, as I mentioned earlier. With the APC channel, you can then ultimately detect the captured exosomes as shown in the third plot. And as you can notice here, a 3-dimensional diagram would be required to show the signal intensity for each bead type separately. But in the PE APC diagram, up to 5 bead types have the same PE signal intensity, and therefore they appear overlapping in the diagram. Nevertheless, with the multiplex bead-based EV assay for flow cytometry, you can easily detect and semi-quantify 37 markers on your EV sample. The advantage of this unique multiplex bead platform is therefore that it allows you to profile proteins on EVs very fast and simple by flow cytometry. The assay works on any flow cytometer with a blue and red laser to detect FITSI, PE, and APC, and it does not demand extensive operator expertise. Thus, it can be easily implemented into most laboratories for routine EV analysis, and you also save precious material by directly screening 37 surface markers simultaneously, and a list of these markers is shown here on the right. So these include common EV markers like the tetraspanin shown here in orange, but also immunology and cancer epitopes can be analyzed. So we also have here, for example, CD20 or CD4, which are general B-cell or T-cell marker respectively. As mentioned, it's a semi-quantitative method, so the signal intensity detected with detection antibodies depends on the EV concentration, and the range of detection was, for example, assessed here by using 5 different doses of EVs isolated from hex cells as input. And as expected, the signal intensity for positively stained bead population decreased with decreasing EV input, as you can see from left to right. However, reliable results were obtained when the supernatant contained about 1 million to 100 million HEK-derived EVs. And taken together, the results show that the bead-based EV assay enables a sensitive semi-quantification of EV surface markers. The assay is also customizable and can be adapted to individual needs. This was shown in the following study, when antibody against FOLR1 was used as staining reagent. FOLR1 on EVs was reported to shuttle folate into the brain and is examined as a biomarker for ovarian cancer. EVs from pre-cleared cell culture supernatant of a common human ovarian adenocarcinoma cell line showed robust expression of abundant markers like tetraspanins and epithelial markers like CD326 when a detection reagent cocktail was used, as you can see in the left plot. When the anti-FOLR1 APC antibody was, however, used as detection antibody, also signals for the CD9, CD63, CD81, and CD326 capture bead populations were gained. This indicates that the EVs express FOLR1 and that those EVs also co-express the tetraspanins and CD326. The data also highlights that custom detection antibodies are compatible with the assay, and therefore it is easily possible to assess whether a certain candidate surface marker that's not detectable by the kit is actually present on EVs or not. The assay is further flexible, so additional antibodies can be added for multiparameter analysis, and no pre-enrichment is necessary to run the Multiplex exosome kit, so EVs can be analyzed by flow cytometry directly from different sample types or after isolation. This is shown here, where the EV surface signatures of major human body fluids like plasma, serum, and CSF were profiled. Briefly, different EV input amounts of each sample were analyzed, and the results were then visualized as heat maps for comparison. And you can see that high and consistent levels of tetraspanins were detected in pre-cleared CSF samples, but also in non-chromatography purified plasma as well as in serum, with no major difference among EV surface marker profiles in the later 2 samples, as you can see in the left heat map. Therefore, under standardized condition, the Multiplex bead-based assay not only represents a valuable tool to characterize, but also to compare EVs from different sources. After learning more about the basic principle of the Multiplex bead-based EV assay for flow cytometry and its features and advantages, let's have a closer look at some data. As my colleague highlighted in the beginning, cancer-derived EVs have great potential to be biomarkers for early clinical diagnosis and evaluation of cancer therapeutic efficiency. In the study depicted here, surface proteins of EVs captured directly from assays obtained from 5 ovarian cancer patients were analyzed with the bead-based assay. Ovarian cancer is among the most common types of cancer and one of the most lethal gynecological malignancies. The low survival rate and poor quality of life of patients with ovarian cancer is in part due to the lack of early diagnostic methods and high chemoresistancy rate. Thus, there is an urgent need to identify new biomarkers for early diagnosis and prognosis of ovarian cancer. And the results of the bead-based assay show that besides the common exosome markers like CD9, CD63, and CD81, also markers were detected indicating blood cell-derived vesicles, which include CD4 and CD8 for T cells and also CD14 for monocytes. Interestingly also was the observation that samples from different patients showed significant differences in the levels of key tumor markers such as CD326 or APRIM and CD133 or PROM1. These results clearly indicate the presence of tumor-derived EVs in the assays of patients with ovarian cancer and are in line with other studies showing that the formation of malignant acids is a hallmark of advanced and metastatic ovarian cancer. Other experiments with the multiple flow assay showed that EVs isolated from cancer cell lines can be distinguished by differing tumor markers. As you can see here, MCSP-positive EVs were captured from all 3 melanoma samples. and MCSP is a dominant melanoma cell surface marker and a well-established target for melanoma immunotherapy. In contrast, melanoma EVs hardly showed any CD326 or EpCAM compared to colon cancer EVs, as demonstrated by the binding to the respective capture beads and highlighted with the arrow in the graph. Thus, you basically have two markers which would allow you to distinguish between the melanoma and colon cancer-derived EVs. I hope these experiments could give a glimpse of the potential the multiplex bead-based EV assay holds. However, many more studies are published employing the assay to investigate the biology of EVs and to explore their potential in clinical applications. And one application I would like to highlight here at the end is the use of the assay to characterize and assess the quality of EVs for clinical use. Especially mesenchymal stem cell-derived EVs have gained a lot of attention as a cell-free component to potentially treat different diseases and conditions like, for example, myocardial infarction or COVID-19. However, ensuring consistency and good quality of the EV therapeutics is of tremendous importance to safely move forward into the clinics. Thus, assessing the general repertoire of 37 surface markers on EV can add an additional level of information for quality control. To sum it up, EVs are membrane-surrounded particles that are secreted by virtually every cell type. They function as intercellular messenger and hold great promise for diagnostic and therapeutic applications. We here at Miltenyi offer a complete workflow for exosome research, from isolation to analysis. The immunomagnetic isolation allows high-purity EV purification and enables high-quality downstream analysis. Additionally, you can physically separate different EV populations from a complex sample. The bead-based flow cytometry assay then enables to screen multiple potential markers on EVs in parallel. And our solutions are widely used by experts in many research fields including immunology, cancer, neuroscience, stem cell research, or for example infectious disease. They can be easily integrated into any workflow to advance both basic and clinical EV research. And with this, we are already at the end of the webinar, so thank you very much for your attention, and we look forward to your questions.
And thank you, Ariadna and Laura, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. And it looks like we already have some incredible questions coming in from our audience members. Our first question: what is the size range for the magnetic isolation beads? And the graphics show them as small compared with EVs, but is this really true with respect? And I think that you have muted yourself. I want to.
Yes, so indeed, the graphics might be a little bit misleading here. So our microbeads are in the size range of 80 to 120 nanometers, so it's pretty much the same size as the exosomes. The advantage of the small size is that the beads stay in suspension, and therefore you have a very efficient binding of the vesicles to these beads because they are in close proximity during the binding reaction.
Thank you so much. And our next question: do you have a release antibody conjugated with beads for EV?
So far, we don't have release antibodies for the exosomes, so this is something we are working on. So the current setup of the beads, they are not releasable, so the vesicles stay on the beads. But you can elute the vesicles from the beads if you lyse the beads. So for RNA analyzer, protein analysis, you can do the lysis on the column, then the beads stay on the column, and you lyse just the protein and RNA content of the beads during the elution, and then go for a downstream analysis, and then you can exclude the beads.
Thank you so much. Now, when using flow cytometry to analyze EVs, is it known that the equipment must be properly rinsed before use? And what cleaning protocol do you use in your flow cytometers?
That's a very good question. So I think this is one of the advantages of using the beads for the analysis of the vesicles. So these are large beads, about 4 micrometers in diameter, so they can easily be detected in flow cytometer like usual cells. And we do not detect single vesicles by this assay. So therefore, if there would be some contamination of vesicles in the solution, we simply would not detect them because we just detect the signals which are related to the beads, and we don't see the signals from the background. So therefore, we have a rather specific signal, and of course, you do the usual cleaning routines you would do for a cell analysis on the flow cytometer. So the same cleaning as you do in your routine work on the flow cytometer. So there's no special cleaning required because we do not analyze single EVs.
Thank you so much. And I want to thank our audience for these great questions coming in. What is the size range for the magnetic isolation beads? And the graphic shows them as small compared to EVs. Is this really true with respect to exosomes?
So I think I already answered this. So the size range is 80 to 120 nanometers. So it's similar to the exosome size range. So it's a little bit misleading in the pictures shown in the graph. So they are more or less as small as the vesicles.
Thank you very much. Do capture beads rely on tetrapanin expressions only, or can they be customized to use other capture strategies?
I think I can answer this question. So our isolation microbeads, so far, we only have products that rely on the tetrapanin expression, CD9, CD81, CD63. But we do not have at the moment other microbeads for exosome isolation. However, we have in the portfolio many other microbeads that could potentially be used for exosome isolation, or at least tested for other markers.
Thank you very much. And what is the maximum number of co-expressed markers that can be detected per?
So again, a very good question. So as you have seen in the presentation, we are applying a kind of ELISA principle. So if we have one capture antibody coupled to the beads, and then a second antibody carrying the label to label the bound vesicles. So you only get a signal if both markers are present on the vesicles. So basically, we always detect the presence of two markers on the same vesicle to get the signal. And of course, as we are using one dye in the standard assay, we always detect two markers on the same vesicle. However, it's of course possible if you're using flow cytometer to use additional dyes, additional colors as another antibody, and thereby you can get more information about additional markers. But for each marker, you always get the information of the two markers present on the same vesicle. So if you stain with two different antibodies, of course, you will not know whether these antibodies stain on the same vesicle or on different vesicles, because, of course, on one bead, many vesicles can be bound. So therefore, it's in principle possible that the staining comes from two different vesicles, giving the same signal, giving a signal on the same bead type. So therefore, you always can detect two markers on the same vesicle. But you can combine this with different markers, of course, to get more information on these vesicles.
Thank you very much. And can you give an example of an infectious disease?
Yes. So yeah, I think currently the most prominent one that's being explored is to treat COVID-19 as an example for infectious disease, be it being used as a vaccine delivery system or to even treat it.
Thank you very much. Now, what do you think about aldehyde sulfate latex beads for analysis of exosomes or flow cytometry?
So these beads are a perfect tool to analyze single markers on the vesicles. So you basically non-specifically bind the vesicles to the beads, and then this allows you again to use standard flow cytometry to detect the markers on the vesicles. However, as said, the binding to the beads in this case is a non-specific binding. So therefore, you of course cannot be sure what you have captured on your beads. So I think having the ELISA principle increased the specificity of the assay. And in addition, due to the color coding of our beads, you can address 37 markers in parallel, and this saves you a lot of time. And also, if you're talking about precious body fluids, also material, because basically you're using the same amount of material and get 37 information about 37 markers instead of just one, using the classical non-specific binding to the beads.
Thank you very much. And again, I want to thank our audience for these great questions coming in. Our next audience member says, thank you very much for this informative talk. I'd like to know if it is possible to isolate exosomes from the supernatant of the fibroblast cell culture.
Yes, in theory, you can isolate exosomes or other EVs from cell culture supernatant of any kind of cell that you're cultivating. This includes fibroblasts or even neurons, MSCs.
Wonderful. Thank you very much. Now, is there any EM data using capture beads?
Yeah, there are publications where they use our exosome isolation kits and then they proceed to EM analysis. And yeah, it is possible to see the exosomes.
And how about, do you need to separate magnet beads from exosomes to use WB analysis?
So it's not required. You can simply lyse the vesicles bound to the beads and then perform the gel analysis, so the gel separation, and then do the Western blot. The beads would stay in the pockets of the gel, so they do not interfere with Western blotting. The other possibility is to lyse the cells on the column and then do dilution within the magnet. Then the beads will stay on the magnet, and again, then you can run the gel for a Western blot and do the analysis. So there's no interference of the beads with the downstream analysis using Western blotting.
Thank you so much. We have time for some more questions, a few more. Do you have any microbial specific antibody that could be used for microbial vesicles detection?
So unfortunately, we don't have these. So when we talk about the Multiplex beads, the staining antibodies, they are human specific. So therefore, we have an additional assay for mouse. So the antibodies are really species specific. So therefore, you would not detect any other vesicles from like the microbiome. So that's, of course, on the one hand, a positive effect, because you know that you really have a specific staining. So, for example, if you're using serum in your cell culture, it's always the risk that you get vesicles from the serum in your samples, and this might interfere with your analysis. And we could show that due to the specificity of the antibodies, we do not get any signals from these bovine EVs. So it's really specific. But on the other hand, if you are interested from different species, the assay will not work, unfortunately.
Thank you very much. Our next question, the LDL share the size of exosomes. Do you know if the LDL express CD9, CD81, or CD63? If this question is affirmative, how can we differentiate this particle?
Again, very good question. So these lipids, they are not associated with the tetraspanin proteins, so therefore they will not be enriched using our microbeads. So in contrast to the physical isolation methods like ultracentrifugation, where you always have the risk that some of these particles have the same density or size and therefore are precipitating together with the vesicles, using the surface markers, you can really get rid of these lipid particles and enrich for vesicles.
Thank you so much. Is it possible to customize the magnetic beads with streptavidin to capture biotin antibodies against specific cell-derived EV markers?
So I can give a little bit of information here, and then maybe Stefan, you can elaborate a little bit on that. I think it is potentially possible to customize or to use streptavidin microbeads to capture EBs that are labeled with biotin.
Thank you so much. And we have time for 2 more questions. Can the automax of Miltenyi be used for immunomagnetic isolation of exosomes?
I think we do not recommend to use the automax for exosome isolation, since it is designed for cell separation. But we have the Multimax M96 separator that it could potentially be used for exosome isolation. We have validated the isolation with this separator here in Miltenyi, and it works fine.
Thank you so much. A final question that we have time for. Are there any special requirements on the flow cytometer to be used for the Multiplex assay?
So there are no special requirements. So most of the flow cytometers can be used to analyze the Multiplex assay. So you need a blue laser, so 488 nanometers, and the red laser, 635 nanometers. And then, so the ability to detect the FITC, PE, and the APC in 3 different channels. So I think that's a setup which most of the flow cytometers can work with. So there might be some exemptions, but most of the flow cytometers can be used, and then you can easily set up the assay on this flow cytometer.
I want to thank all 3 of you for your time today. Would you like to provide any closing remarks before?
No, I thank you very much for all these interesting questions. Yeah, and you can find many more information on our website, or reach out to us or to our Miltenyi colleagues for more information.
Yeah, also thank
you again for your presence. Oh, go ahead. Please go ahead.
Yeah, thank you also from my side for all the questions. It was really an interesting discussion, and unfortunately we could not answer all the questions. But for those questions we could not answer, we will come back to you per email and answer these questions as well.
Yes, exactly. So
Absolutely.
also from my side, thanks a lot for this lovely discussion.
I want to thank all 3 of you again for your time today and for your important research. And I also want to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. And before we go, I want to thank the audience for joining us today for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.